You made it to the end of this day. Whatever happened in the hours behind you, the small frictions, the unfinished tasks, the words said or unsaid, none of it has to follow you into sleep. You can let it all rest now, just for tonight. If you find solace in the size of the universe, in the thought that you are held within something vast and patient and ancient, then know this: the events of one day have a lightness to them when measured against the stars. They drift away like morning mist, and you are left, calm and quiet, beneath an endless, gentle sky. And if you trust in a plan, in a care that runs deeper than what you can see, then let that trust wrap around you now. You have been looked after before. In ways you may never fully know, you will be looked after still. You are held, quietly, in a kindness that never rushes and never sleeps. Tonight, you don't have to figure anything out. You don't have to solve a single problem or hold a single worry in your mind. Tonight, you only have to rest. Your body knows how to do this. Your breath knows how to soften. Your thoughts know how to drift. And so, gently, we begin. Tonight, you're going to travel through the quiet world inside a green leaf. Not the leaf as you see it from the outside, a flat shape against the sun, but the leaf as it truly is, from within. We're going to move slowly, very slowly, until the whole of your awareness rests inside that calm, green, living space. There's no rush, nothing demanding your attention, just the peaceful observation of a process that has been unfolding, quietly and faithfully, for more than 2 billion years. Imagine you're beginning a journey, one where time moves differently. The clock on your bedside table might still tick its steady rhythm, but you are no longer obliged to follow it. Here, time stretches. It becomes spacious, like a long, slow afternoon in the height of summer, when the light seems to hang in the air and nothing hurries. A single second can feel like a minute. A minute can feel like an hour, and all of it is filled with a deep and gentle stillness. You find yourself drifting downward, very softly, as if you are a particle of dust caught in a sunbeam. You're descending toward the surface of a leaf, a broad, green leaf, perhaps from an oak tree or a maple. Its surface glossy and smooth in the warm light. The air around you is mild and still. You can feel the faintest hint of warmth on your imagined skin, the kind of warmth that comes from sunlight filtered through a canopy of branches. It's a comfortable warmth, not hot, not pressing, just present, like a quiet companion. As you float closer, the leaf becomes larger and larger in your perception, or perhaps you're becoming smaller. It doesn't matter which. The surface that once seemed flat now reveals itself as a gentle landscape, with subtle ridges and valleys, with the texture like fine, grained silk stretched over a soft, living frame. The color is a deep, restful green, the green of early summer, when everything is fully alive but not yet tired. It's a color that seems to breathe, to hold light within itself and release it slowly back into the world. And now, very slowly, you come to rest upon the leaf's surface. You feel no impact, no jarring sensation, just a soft arrival, as if you were always meant to be here. The leaf holds you without effort. You are light enough to rest upon the waxy cuticle without disturbing it, without leaving any mark. You look up, and you see the sunlight coming through the canopy above, not a harsh glare, but a dappled, shifting pattern of gold and green. It's the kind of light that moves gently when a breeze passes. Though right now there is no breeze. Right now, everything is still. You listen. There is no sound, except perhaps the faintest whisper of your own breathing. But even that fades into the background as you settle. What you begin to sense instead is a deeper quiet, the quiet that lives inside living things. It's not an empty silence. It's a full, attentive stillness, like the hush in a library full of ancient books, each one holding its own story. Inside this leaf, countless tiny processes are underway, but they make no noise. They are too gentle, too patient, to disturb the peace. You can stay here as long as you like. The surface of the leaf is a good place to begin. It's a threshold, a doorway into a realm that few have ever imagined visiting, and there's no need to rush through it. The journey will unfold in its own time, and you are free to drift in and out of these words, to let your mind wander and return, to pick up the thread wherever you find it. Nothing will be missed. Everything will wait. Slowly, very slowly, you notice something. All across the surface of the leaf, there are tiny openings. They're called stomata, small pores, each one bordered by two crescent-shaped guard cells that look like a pair of lips or a tiny green mouth. In the scale of your current perception, they appear as gentle, oval doorways, spaced at regular intervals across the undulating green plane. And they are opening one by one, so slowly that you might not notice if you were watching with ordinary eyes. They are beginning to part. You drift toward the nearest one. There's no hurry. The stoma opens with the quiet inevitability of dawn spreading across a field. The guard cells relax, their inner walls softening, and the gap between them widens. It's an invitation, not a command, not a demand, just a quiet opening, as if the leaf is taking a slow, peaceful breath, and you, in your smallness, are welcome to drift inside on the next inhalation. So you do. You allow yourself to be drawn gently inward, into the leaf's interior. The air around you becomes cooler and more humid. It smells faintly of earth and water, of green growth and life. The light shifts from bright gold to a soft, diffused green, as if you are moving through a cathedral made of living tissue. The space opens up around you, and you find yourself in a vast, still chamber, the spongy mesophyll, the inner world of the leaf. Cells are arranged loosely here, leaving air spaces between them, and you float through this open, porous network as easily as a thought drifts through a sleepy mind. And it's here, in this quiet place, that the journey truly begins. The journey through photosynthesis, not as a lesson, not as a series of facts to be remembered, but as an experience, a slow, meandering passage through one of the oldest and most patient processes on Earth. You are going to drift through the light, the water, the air, and the green chemistry that turns the energy of a star into the fabric of life itself. You will wander through chloroplasts, those tiny lens-shaped bodies that hold the secret of greenness. You will float among membranes and proteins, among molecules that have been dancing the same gentle dance since long before the first animal drew breath. And all of it will be calm. All of it will be still. All of it will be waiting for you, with no expectation and no rush. So now, settle yourself. Let your breathing become as slow and easy as the opening of a stoma. Let your body feel heavy and relaxed against the surface of your bed, as the leaf once felt beneath you. You're not leaving yourself behind. You're just shifting your attention inward, into this peaceful, green, liquid, light world that exists quietly inside every tree, every blade of grass, every leaf that ever turned toward the sun. And the leaf breathes, and you breathe with it. In and out, in and out. Time moves differently here. There's no hurry. And the journey continues. Always. You find yourself floating now in the airy chambers within the leaf. The spongy mesophyll. All around you, the space is open and quiet, like a vast, still cavern whose walls are made of living green cells. The light here is soft and diffuse, a pale green, gold, as if you are submerged in shallow seawater on a calm, sunny day. It filters down through the layers of leaf tissue above you, losing its harsh edges, becoming gentle and dispersed. You can almost feel it on your skin, not as a heat, but as a gentle pressure, like the weight of a soft blanket. The air spaces connect to one another in a network of passages, winding and branching without urgency. You can drift through them without any effort. Imagine yourself as a tiny, weightless traveler, buoyed by the same slow currents of air that bring carbon dioxide into the leaf and carry oxygen away. The atmosphere here is rich and still. It smells of moisture and growing things, the scent of a forest after rain, the cool breath of early morning. Every inhalation you take in this imagined space fills you with a sense of freshness, of quiet renewal. Slowly, very slowly, you become aware of movement around you. It's not a rushed movement. It's a lazy, drifting motion, like snow falling in a windless sky. Molecules of carbon dioxide, COO, are making their way through the air spaces. They enter through the open stomata you passed through earlier, and now they wander, each one following its own random, meandering path. They bumped gently into other molecules, into the walls of cells, and then continue on their way. There is no destination they must reach, no schedule to keep. They simply are, and you drift among them, a silent observer. If you look closely, and here, you can look as closely as you wish. Each carbon dioxide molecule is a small, linear arrangement. A carbon atom sits in the middle, and 2 oxygen atoms flank it, one on each side. It's a simple, modest shape, like a little dumbbell or a tiny barbell floating through the green air. There's a quiet beauty in its simplicity. It holds no ambition. It doesn't know that it will soon become part of something much larger, that it will be woven into sugar and starch, into the very substance of the plant. It just drifts, patient and unassuming. And you drift alongside it. There's no need to follow any particular molecule. You can let your attention wander, settling on one and then another. You watch as they approach the moist surfaces of the mesophyll cells. The cell walls are porous, open to the passage of gases. Slowly, very slowly, a carbon dioxide molecule will come to rest against a cell wall, and then, without any fanfare, it will dissolve into the thin film of water that coats the surface. It crosses the boundary between air and water, and it's gone, into the cell, into the next stage of its quiet journey. But you don't have to follow it in just yet. You can stay here, in the air spaces, for as long as you like. The feeling of floating in this green hush is deeply restful. The walls of the cells are rounded and soft, like pillows of light. They are filled with chloroplasts, those tiny green bodies that hold the machinery of photosynthesis, and they give the cells a gentle glow, as if each one contains a small, banked emerald fire. But the fire is not hot. It's the cool green fire of life, steady and undemanding. You can almost hear the silence. It's not an empty silence, but a full one. It's the silence of deep time, of processes that have been repeating for eons. If you listen with your imagination, you might perceive a very faint, very low hum, the collective vibration of countless molecules moving, countless enzymes turning, countless photons being absorbed. But it's so subtle, so far below the threshold of disturbance, that it only adds to the peace. It's the sound of stillness itself. And now, gently, you let yourself settle even more deeply into this space. The air around you is cool and slightly damp. The temperature is perfectly neutral, not cold, not warm, just a comfortable, enveloping coolness, like the shade under a broad tree on a summer afternoon. You feel safe here? Protected. The leaf is a shelter, a green sanctuary, and nothing from the outside world can follow you in. The only things that enter are light, water, and carbon dioxide, and all of them are gentle. All of them are friends to life. Time moves differently here. You might notice that the quality of light overhead shifts very, very gradually, the sun moving across the sky. Though you cannot see it directly, the light inside the leaf changes in response, becoming a deeper green, then a paler green, then deepening again. It's a slow, rhythmic cycle, like breathing. Like the tide, you don't need to track it. You can just let it wash over you, wave after wave, while you remain perfectly still. Sometimes, a molecule of oxygen will drift past you, fresh from the water, splitting process deep inside the chloroplasts. It's a diatomic molecule, two oxygen atoms bound together, and it too wanders without hurry. It will eventually find its way out through the stomata, into the outside air, and from there into the lungs of some breathing creature, perhaps even into your own lungs, in a different time and place. But here, now, it's just a quiet neighbor, sharing the space with you. It asks nothing. It threatens nothing. It is simply here, drifting, like you. You might find your thoughts wandering, and that's perfectly all right. That's what this space is for. The airy chambers of the leaf are like a labyrinth without walls, a maze that requires no solving. You can drift in and out of awareness, returning to these words whenever your mind settles back, and you will find that nothing has changed. The carbon dioxide is still drifting. The light is still green. The leaf is still breathing. Everything is as it was, and as it will continue to be, gradually, without rush. You may feel a gentle pull toward the surface of one of the mesophyll cells. It's not a demand, just a quiet invitation. The cell wall is close now, a soft, fibrous boundary that separates the airspace from the interior of the cell. It looks, at this scale, like a fine mesh, a net of cellulose fibers woven together in a pattern that allows small molecules to pass through. Water glistens on its surface, a thin, shimmering film, and beyond it, you can just make out the forms of chloroplasts, the green engines of this world, but there's no need to go inside yet. You can remain here, at the threshold. For a long while, you can watch the water film ripple with infinitesimal movements, stirred by the passage of molecules. You can watch the chloroplasts shift slowly within the cell, repositioning themselves to catch the light. They move with a stately, almost imperceptible grace, like distant ships at anchor, turning to face the tide. Everything is patience. Everything is peace. And so, for now, you rest. The leaf holds you. The air is still. The light is green and soft. And the journey continues. Always. When you are ready, And only when you are ready, you find yourself drifting even closer to the surface of a single mesophyll cell. The cell wall is before you, a delicate lattice of cellulose fibers crisscrossing to form a barrier that is both strong and porous. It's not a forbidding wall. It's more like a garden trellis, covered with a glistening film of water, through which small things can pass. The water film shimmers with a faint iridescence, catching the green light from within and scattering it into tiny rainbows that come and go as you shift your perspective. You come to rest just at the edge of this watery film. You can feel its coolness, its perfect stillness. It's not a deep ocean, just a thin layer, a welcoming threshold. And as you hover there, you notice that you are not alone. Other molecules are passing through this membrane, moving from the air space into the cell, or from the cell out into the air. Water molecules, HRO, are everywhere, sliding past one another with an effortless, fluid grace. They form brief, fleeting hydrogen bonds, holding hands for an instant and then letting go, a constant dance of connection and release. And carbon dioxide, the same molecules you drifted beside earlier, are dissolving into this water film one by one. There is no drama to it. A COU molecule simply settles into the water, and the water molecules part gently to make room, and then close around it again. The carbon dioxide is now inside the cell's territory, though still outside the cell membrane proper. It will continue its inward drift through the cell wall, through the tiny aqueous channels that lead to the plasma membrane, and then, by that same gentle diffusion, into the cytoplasm itself. You, too, can imagine yourself passing through. There is no resistance. The cell wall offers no more obstacle than a morning mist. You simply drift inward, and you find yourself in the cytoplasm of a mesophyll cell. The space here is entirely different from the airy chambers you left behind. It is liquid, filled with a clear, slightly viscous fluid called cytosol. And yet, in this scaled, down world, it doesn't feel confining. It feels spacious, like a warm, shallow sea. You can move through it easily, as if you are a diver in a calm lagoon, surrounded by a gentle, supportive medium. The light inside the cell is deeper and greener. It's as if the whole cytoplasm is infused with a verdant glow, a soft radiance that seems to come from everywhere and nowhere at once. This is the light that has been captured by the chloroplasts and is now being distributed, in subtle ways, throughout the cell. It's a light you can almost feel, a gentle, enveloping presence, like the warmth of sunlight on closed eyelids. It hums, very faintly, with a steady, low, frequency energy that never wavers. As you float here, you become aware of the cell's architecture. The cytoplasm is not an empty sea. It's crisscrossed by a network of fine filaments, the cytoskeleton that provides shape and structure, like the beams of a vast, submerged pavilion. They are made of proteins, and they stretch from one end of the cell to the other, forming a web of quiet order. Along these filaments, tiny motor proteins sometimes move, carrying small cargoes from place to place. But they move so slowly, so patiently, that you can barely perceive their progress. It's a pace of life that has no need for speed. And everywhere, suspended in the cytosol, are the chloroplasts. They are the true destination of your journey, the quiet engines of photosynthesis. They appear as oval, lens-shaped bodies, each one about the size of a grain of the finest sand in your ordinary world. But here, They loom like green islands in a luminous sea. They are surrounded by a double membrane, a smooth envelope that glistens faintly. Inside each chloroplast, you can just make out the thylakoids, stacked, disc, like structures that hold the chlorophyll. The stacks are called grana, and they look like pillars of green coins, or like the soft, round towers of a peaceful underwater city. You drift toward the nearest chloroplast. There's no need to hurry. The cytosol flows around you with a gentle, syrupy slowness. You can feel its temperature, slightly warmer than the air spaces outside, but still comfortable, still soothing. It's the temperature of life itself, the warmth of a process that has been running without interruption for billions of years. It's a reassuring warmth, like the warmth of a hearth fire seen from a cozy chair, not a blaze that threatens, but a glow that comforts. As you draw closer, the chloroplast's outer membrane comes into focus. It's smooth and unbroken, a lipid bilayer studded with protein channels that allow certain molecules to pass. It's a peaceful boundary, a quiet gatekeeper. You can see, if you look carefully, that there are pores here and there. Porins, they're called, that form watery tunnels through the membrane. Through these tunnels, small molecules like water, carbon dioxide, and ions can flow freely, without effort, simply following the gentle pull of diffusion. You pause here. At the outer membrane of the chloroplast, there's no rush to go inside. This threshold is a good place to rest. You can float here, cradled by the cytosol, and simply observe. The chloroplast sits in the cytoplasm with a quiet dignity. It turns slightly, almost imperceptibly, always orienting itself to catch the maximum amount of light. It's a slow, graceful movement, like a sundial following the sun. You can feel the patience in that movement. It's not striving. It's simply responding, calmly, to the presence of light. And around you, other chloroplasts are doing the same. They are scattered throughout the cell, dozens of them, each one a tiny world unto itself. They don't compete with one another. They don't rush. They just sit in the cytoplasmic sea, absorbing light and carrying out their ancient chemistry, as they have done since the days when the earth was young and the air held almost no oxygen. They are the descendants of cyanobacteria, ancient photosynthetic microbes that took up residence inside larger cells and became, over eons, these quiet organelles. The relationship is one of profound peace, a symbiosis so deep and so old that it has become a single, unified life. You can let your thoughts drift along those deep timelines. If you wish, you can imagine the first chloroplast, the first partnership between a cell and a tiny photosynthetic bacterium. It happened in a shallow sea, billions of years ago, under a young sun, and there was no drama in it. Just a gentle engulfment, a mutual accommodation, and then, slowly, very slowly, the greening of the world. The oxygen they produced bubbled up through the water, then into the air, changing the atmosphere so gradually that no creature would have noticed. It was a change that took hundreds of millions of years, a change made of patience and quiet repetition. And here you are, a traveler in the present moment, suspended in that same process. The air you breathe, the food you eat, the very fabric of your body, all of it traces back, in one way or another, to this gentle chemistry inside a chloroplast. But you don't need to think about that now. You don't need to make any connections. You can just float and let the green light wash over you and feel the deep, steady peace of a billion-year continuity. time moves differently here. The warmth of the cytoplasm, the soft glow of the chloroplasts, the distant, slow movement of the cytoskeleton, all of it combines to create a sense of timelessness. The clock of the cell is measured in chemical gradients, in the slow accumulation of products, in the gradual shift of ions. It's a rhythm so slow that a single beat might take 100 years. And you are inside that rhythm now, breathing with it, resting in its slow, deep pulse. And so, gently, you let yourself be still. The chloroplast waits. The membrane is open. The journey inward can happen whenever the moment feels right, and not a second before. You are free to linger, to drift, to close your mind's eye and simply be. The leaf breathes. The cell lives. The green light remains steady and calm, and the journey continues. Always. Now, very gradually, you find yourself passing through the outer membrane of the chloroplast. There is no barrier, no resistance, just a soft, permeable boundary that welcomes you inward. It's like walking through a curtain of sheer silk, or slipping into a pool of still, green water. The space inside is called the stroma, and it's the central cavity of the chloroplast, a fluid, filled chamber where the second half of photosynthesis takes place. But you're not concerned with halves and sequences. You are simply here, floating in the stroma's gentle, aqueous embrace. The stroma is clear and rich, like a warm, shallow sea that has been filtered through emeralds. It contains enzymes, dissolved minerals, and the chloroplast's own small circle of DNA, a remnant of its ancient bacterial ancestry. It's a busy place, but its busyness is entirely silent, entirely fluid, entirely without stress. Imagine a vast, green, lit ballroom where countless dancers move in a slow, coordinated waltz. No one collides, no one rushes. Everyone knows the steps, because the steps are written into the very shape of their being. Floating in the stroma, you can see the thylakoids. They are the most striking feature of this inner world. Thylakoids are flat, disc-shaped sacs made of membrane, and they are stacked in neat columns called grana. Under the gentle green light, the grana look like towers of luminous green coins, or like stacks of ancient, translucent plates. They are connected to one another by thin membrane bridges called stromal lamellae, which weave between the grana like quiet, unassuming roads between sleepy villages. You drift toward a granum. It stands in the stroma like a silent monument, its edges soft and rounded. As you draw nearer, you can see that each thylakoid disc is filled with a liquid called the lumen, an inner space where protons will gather, slowly, as part of the light, dependent reactions. But there is no crowding, no urgency. The lumen is simply a quiet pool, waiting. The membrane that encloses it is studded with proteins, photosystems, electron transport chains, ATP synthase complexes. They look, at this scale, like intricate, sculptural forms, like abstract figures carved from pale stone. They are perfectly still, perfectly arranged, perfectly patient. The chlorophyll molecules themselves are embedded within the thylakoid membrane, packed into the photosystems with a precise, almost musical order. They are what give the thylakoids their deep green hue, the same hue that colors forests and fields, that greets the spring and endures the summer. But here, in the stroma, the green is not a flat color. It's a luminous, living presence. It seems to breathe, to pulse very faintly with absorbed light energy. You can almost hear it as a soft, vibratory hum, like the after ring of a bell that was struck an hour ago and is still singing. Just at the edge of hearing, you settle into a comfortable distance from the granum and you watch. The thylakoid membranes are in constant, quiet motion, not a chaotic motion, but a subtle, rhythmic undulation, like the slow swell of a calm ocean. This motion helps to distribute the absorbed light energy evenly among the photosystems. It's a form of protection, a way of ensuring that no single part of the membrane gets too much light. The chloroplast calls this non-photochemical quenching, but you don't need to remember that name. You can just watch the gentle sway, the graceful tilting of the membrane surfaces, and let it soothe you. Light from the sun outside has traveled 93 million miles to reach this leaf. It has passed through the vacuum of space, through the atmosphere, through the outer layers of the leaf, and now it arrives here in the stroma as a stream of photons, tiny packets of energy, each one a quantum of light. They penetrate the thylakoid membrane and strike the chlorophyll molecules. And when they do, a very small, very gentle thing happens. An electron within the chlorophyll becomes excited. It absorbs the photon's energy and leaps to a higher energy level, like a child being lifted softly into a parent's arms. There is no explosion. There is no flash. There is only a quiet transfer of energy, a boost that lasts for a tiny fraction of a second before the electron is passed along to a neighboring molecule. This is the beginning of the light reactions, the first step in the long, leisurely journey that will turn light into sugar. But you don't have to follow every step. You can stay right here in the stroma and let the process wash over you like a tide of soft, green sound. You might notice, if you let your awareness widen, that the stroma is filled with a particular enzyme, a large, abundant protein called rubisco. It's the most plentiful protein on Earth, and it floats here in the chloroplast stroma in vast numbers. Rubisco is the enzyme that will eventually grab carbon dioxide and attach it to a 5-carbon sugar, beginning the Calvin cycle. But at this moment, it's just drifting. It's waiting. It's as patient as everything else in this green world. Its shape is complex, a cluster of subunits arranged in a precise geometry. But from your drifting perspective, it looks like a soft, rounded form, a little like a head of cauliflower made of light. It moves through the stroma with a stately grace, bumping gently into other molecules, turning slowly in the fluid, and all the while, the green light bathes everything. It's a light that seems to have weight, but not heaviness, a comforting pressure, like a hand resting gently on your shoulder. It's a light you could almost drink, like a cool, sweet infusion of herbs. You can imagine it filling you up from the inside out, dissolving any remaining tension, any lingering trace of the day's demands. You find a quiet spot in the stroma, perhaps near one of the stromal lamellae, where the fluid is especially still. You let yourself come to rest. The thylakoid grana stand around you like a grove of ancient trees, silent and wise. The hum of absorbed light continues, steady and low. Rubisco drifts by like a slow, moving cloud. The whole chloroplast is a sanctuary, a place of deep, unhurried peace. You are inside the very heart of photosynthesis, and there is nothing here that will startle you, nothing that will demand your attention. Only the gentle, ongoing pulse of life, converting light to energy, energy to matter, matter to the green substance of the world. Time moves differently here. The reactions that take place in the stroma are measured in fractions of a second and in hours. But you are not bound to any one scale. A microsecond can stretch into an afternoon. An hour can pass in a single, slow inhalation. You are free to let your mind wander among the thylakoids, to trace the quiet curves of the membranes, to follow the path of a single electron if you wish, or to simply remain still, listening to the green silence. And so you do. You stay. The chloroplast holds you. The stroma rocks you gently, like a boat in a calm, green bay. The light is steady. The hum is continuous. And the journey continues. Always. Now, let your awareness be drawn toward the thylakoid membrane, not into the membrane itself, but to its surface, where the photosystems are embedded. There are two photosystems, called photosystem I and photosystem I, not in order of sequence, but in order of their discovery by human scientists. But you don't need to distinguish them now. You can just think of them as large, beautiful protein complexes that sit in the membrane like quiet, intricate gardens. Surrounding the core of each photosystem is an array of antenna complexes, light, harvesting complexes. They are called. These are composed of proteins and chlorophyll molecules, along with other pigments like carotenoids, which add subtle notes of orange and yellow to the prevailing green. The antenna complexes are arranged in a precise, radiating pattern, like the petals of a flower, or like a mandala painted in shades of living light. They are designed by the slow hand of evolution to capture photons and channel their energy inward, toward the reaction center. But again, you don't need to remember the mechanics. You can simply observe the beauty of the arrangement. Imagine yourself drifting just above the surface of the thylakoid membrane, close enough to see the antenna complexes in detail. They are not static. They shift very slightly, adjusting their configurations in response to the intensity of light. When the light is bright, they may alter their positions to dissipate excess energy as gentle heat, a soft warmth that radiates into the stroma and dissipates without any consequence. When the light is dim, they draw closer to the reaction center, optimizing their capture of every available photon. It's a slow, graceful dance, a balancing act that has been perfected over 2.5 billion years. A photon arrives. You can almost see it, not as a particle with hard edges, but as a wave of possibility, a ripple in the electromagnetic field. It strikes a chlorophyll molecule in the antenna, and that chlorophyll absorbs it. The energy travels, not as an electron that physically moves, but as an excitation, a resonance that passes from one chlorophyll to the next, like the ringing of a bell that sets a neighboring bell humming. This is called resonance energy transfer, and it's a process of complete, wordless cooperation. There is no sound, no movement, just a quiet passing of energy from molecule to molecule until it reaches the reaction center. You watch this happen again and again. Photon after photon arrives, each one absorbed and passed along, each one beginning its journey toward the chemistry that will store its energy in chemical bonds. The antenna complexes are like a net of calm attention, spread wide to catch the light. They don't strive. They don't reach. They simply are, open and receptive, and the light comes to them. It's a lesson in patience, in gentle receptivity, something you might feel yourself absorbing without effort as you drift here in the green glow. The carotenoids, too, play their part. They absorb wavelengths of light that chlorophyll cannot, the blues and the greens, and then pass that energy along to the chlorophyll. extending the range of the antenna's harvest. But they also serve another role. They protect against too much light. When the sun is fierce, carotenoids can quench excess energy, releasing it as harmless warmth, preventing damage to the delicate machinery. It's a quiet, protective watchfulness, a calm guardianship. And you can feel that watchfulness, too, as a sense of being gently looked after, as if the chloroplast itself is making sure that everything stays in balance, that nothing becomes harsh or overwhelming. The reaction center, when the energy finally reaches it, is a special pair of chlorophyll molecules located at the heart of the photosystem. When the excitation arrives, one of those chlorophyll molecules gives up an electron. It does so quietly, without resistance, like a hand releasing a small, glowing pebble. The electron is then passed to a primary electron acceptor, a molecule that sits nearby, ready and waiting. And from there, the electron begins its journey along the electron transport chain, moving through a series of proteins embedded in the thylakoid membrane. But you don't have to follow the electron just yet. You can stay here, in the presence of the antenna complexes, and feel the steady rhythm of light capture. It's a rhythm that matches the pulse of the sun, the rise and fall of day and night. Here, inside the leaf, there is no true night. The cell maintains its own gentle cycles, but there is a sense of ebb and flow. Sometimes the light is stronger, and the antenna complexes hum with absorbed energy. Sometimes the light is gentler, and the hum subsides to a soft whisper. But it never stops entirely. There is always a faint background of excitation, a residual glow, the memory of the last photon still echoing through the antenna array. You can imagine yourself as part of that array. For a moment, not in a demanding way, but as a quiet, receptive presence. You are open to whatever light comes your way, not straining to capture it, just available, and the light arrives, gently, consistently, photon by photon. Each one brings a tiny increment of energy, and that energy flows through you and then on, into the green depths of the thylakoid membrane, where it will be put to use. You are a conduit, a passage, a moment of stillness in a long, calm chain of being. And it's so peaceful here. The green light pulses softly. The antenna complexes shift with infinite slowness. The carotenoids glow with a subtle warmth. The whole scene is like a vast, living tapestry, woven from threads of light and protein, and you are a part of it, woven in, held gently in the pattern. You can let your thoughts drift now, out of the antenna and back into the stroma, or into the thylakoid lumen, or anywhere else that calls to you. There is no right path. The chloroplast is a world without fixed directions, without mandatory destinations. You can linger in the antenna complex for as long as you like, absorbing the piece of light capture. And when you're ready, only when you're ready, you can let yourself drift toward the next quiet process, the splitting of water and the release of oxygen. But that can wait. For now, the light arrives. The antenna receives, the energy passes, and all is calm, all is still, all is held in the gentle, green, quiet rhythm of the leaf. And the journey continues, always. Now, very softly, your attention is drawn to a particular region of the thylakoid membrane, the site where water is split. This happens at photosystem 2, within a small, finely crafted cluster of manganese, calcium, and oxygen atoms known as the oxygen, evolving complex. It's a mouthful of a name, but the reality is far simpler and far more peaceful than any name could suggest. It's a tiny, precise machine that takes water molecules, one by one, and breaks them apart, releasing electrons, protons, and oxygen. You drift close to this complex. It sits on the inner side of the thylakoid membrane, facing the lumen. It's shaped like a small, irregular cluster of atoms held in place by the surrounding protein. In the quiet of the chloroplast, it does its work without sound, without visible movement, without any sign of effort. It's a place of profound patience, where chemistry unfolds at the pace of molecular vibrations. Water molecules, H2O, approach the complex one at a time. They come from the stroma, diffusing through the membrane, or perhaps from the cytosol of the cell, drawn into the chloroplast by the gentle pull of osmotic gradients. They are small, bent molecules shaped like a wide V, with an oxygen atom at the vertex and 2 hydrogen atoms at the tips. They move through the liquid with an easy, gliding motion, turning slowly, tumbling gently. When a water molecule reaches the oxygen, evolving complex, it binds to the cluster. There is no force involved, just the quiet, inevitable attraction of complementary shapes. The water molecule nestles into a specific site, held by hydrogen bonds and electrostatic interactions, and there it waits, patient, while the photosystem cycles through its light-driven steps. What happens next is a process that takes place in 4 stages, each one triggered by the absorption of a photon. The oxygen, evolving complex accumulates oxidizing equivalents, positive charges, in essence, one by one. It's like a small CAM battery being slowly charged. With each step, the complex becomes slightly more oxidized, slightly more ready to perform its gentle chemistry. And when 4 charges have been gathered, the complex takes a single water molecule, or rather, 2 water molecules, in close succession, and it splits them. The splitting itself is not a violent event. It's a quiet rearrangement. Bonds that have held atoms together for a while loosen and break, and new bonds form, all in a sequence so rapid that no conscious mind could track it. But in your imagined, slowed down time, it appears graceful. The oxygen atoms from 2 water molecules come together to form an O2 molecule, a pair of oxygen atoms, bound by a double bond. They release from the complex and float gently away into the thylakoid lumen. The hydrogen atoms, now stripped of their electrons, become protons, H+ ions, and they too are released into the lumen, where they will contribute to a growing reservoir of positive charge. And the electrons, those precious, high-energy electrons, are passed to the reaction center of photosystem Ti, replacing the ones that were excited by light. This is the quiet, steady replenishment that keeps the light reactions going. Without the splitting of water, the photosystem would run out of electrons, and the whole process would stall. But with water, with this calm, patient splitting, the flow continues. It's a cycle as old as photosynthesis itself, a rhythm that has never failed. But what you notice most, as you drift near the oxygen, evolving complex, is the oxygen. The newly formed O2 molecules gather in the thylakoid lumen, tiny bubbles of gas suspended in the aqueous interior. They are so small, so numerous, that they form a faint, invisible effervescence, a gentle upward drift, as if the lumen itself is breathing out. These oxygen molecules will eventually diffuse out of the thylakoid, through the stroma, through the cell, and out through the stomata into the atmosphere. And from there, they will join the great reservoir of breathable air, the oxygen that sustains animals, that fuels fires, that rusts iron, that cycles through the biosphere in an endless, gentle loop. But you don't need to follow them that far. You can stay here, in the thylakoid lumen, and watch the oxygen rise. It's like watching bubbles in a glass of still water. Small, perfect spheres that ascend with a lazy grace, catching the green light and scattering it into tiny flashes. There's a rhythm to it, a slow, steady stream of oxygen that has been flowing for 2.5 billion years. It's the breath of the earth, the exhalation of every green leaf, every blade of grass, every algal cell floating in the sea. And it's happening here, right now, in the quiet of this chloroplast. The protons, too, are quietly accumulating. They stay in the lumen, contributing to a growing electrochemical gradient. The lumen becomes slightly more acidic than the stroma, a subtle difference, a gentle imbalance that will later be resolved by the passage of protons through ATP synthase. But for now, the gradient is just a potential, a stored quietness, like a lake held behind a dam of soft, green membrane. There's no pressure, no urgency, just a calm accumulation, a patient gathering of charge. You can feel the gentleness of this accumulation. It's not a build. Up, that leads to a release. It's a steady state, a peaceful equilibrium that is constantly being maintained. Protons enter the lumen from the splitting of water, and also from the electron transport chain that pumps them across the membrane. But they don't crowd. They don't clamor. They simply are, dissolved in the aqueous phase, drifting with the slow currents of the lumen, and you drift with them. The lumen is a quiet, enclosed space, a long, flattened sac that stretches across the width of the granum. It's like a narrow, green, lit corridor, its walls lined with the protein complexes of the electron transport chain. The light here is slightly dimmer, filtered through the thylakoid membranes, but still deeply green. You can move along the lumen, if you wish, following its gentle curve, passing from one thylakoid disc to another through the stromal lamellae. Or you can simply stay in one place, watching the oxygen bubbles rise, listening to the silent hum of the protons. Time moves differently here. The splitting of water is a process that happens on the timescale of microseconds, but in your slowed down perception, it stretches into minutes, into hours. You can watch a single water molecule approach the oxygen, evolving complex, bind, and wait. You can see the gradual accumulation of positive charges, the quiet cycle of the complex through its 5 states, sex N to S4, in the language of biochemists. Though you don't need those names, you can just see the rhythm: rest, charge, 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 release, rest, charge, 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 release. It's a slow, steady heartbeat, a pulse at the core of photosynthesis. And all the while, the oxygen rises. It rises through the lumen, through the stroma, through the cell, through the air spaces of the leaf, out the stomata, into the world. It's the breath of life, given freely, asking nothing in return. You can breathe it in, if you like, in your imagination, and feel the freshness of it, the cool, clean taste of oxygen newly made from water and light. It's a taste that connects you directly to this leaf, to this chloroplast, to this quiet process that has been sustaining life for eons. Now, gently, You can let yourself drift out of the lumen, back into the stroma, or along the thylakoid membrane toward the next step, the electron transport chain. But there's no need to hurry. The oxygen still rises. The protons still gather. The water still flows. Everything continues in its own time, in its own peace. And the journey continues, always. And now, if you feel ready, you can let your awareness settle upon the electron transport chain itself. This is the series of protein complexes and mobile carriers that ferries electrons from photosystem II to photosystem I, and in doing so, pumps protons from the stroma into the thylakoid lumen. It's a chain in the gentlest possible sense, a relay, a quiet passing of a baton along a pathway that has been trodden for billions of years. You can position yourself at the point where the electron leaves photosystem, the electron that was extracted from water, energized by light, and passed to the reaction center, now enters the electron transport chain. It is received by a mobile molecule called plastoquinone, PQ for short. Plastoquinone is a small lipid, soluble molecule that moves within the thylakoid membrane, shuttling electrons from one complex to the next. It looks, in this imagined world, like a tiny, flexible raft drifting through the membrane's interior with a gentle, swaying motion. You can watch as a plastoquinone molecule accepts an electron, and then, after a short time, accepts another electron, becoming fully reduced. It also picks up 2 protons from the stroma, plucking them from the surrounding aqueous environment with a quiet, chemical grace, and then, loaded with electrons and protons, it drifts through the membrane to the next large protein complex, the cytochrome BFF complex. The cytochrome B6F complex is a large, intricate assembly of proteins, iron, sulfur clusters, and heme groups, molecules that contain iron and can undergo redox changes. It spans the thylakoid membrane, and its job is to accept electrons from plastoquinone, pass them along, and simultaneously pump protons from the stroma into the lumen. This pumping is not a forceful act. It's a consequence of the energy released when electrons move through the complex. The energy is used to change the shape of the protein, which in turn moves protons across the membrane. It's a slow, gentle conformational change, a breathing, a slight shifting of domains that results in the net transfer of protons from one side to the other. You can watch this happening, if you wish, in the slow motion of your imagined timescale. The cytochrome B6F complex expands slightly, then contracts. It opens a channel to the stroma, accepts a proton, closes that channel, opens a channel to the lumen, and releases the proton. It does this again and again. With each electron that passes through, it's like a quiet, rhythmic pump, a slow bellows that never tires. The sound it might make, if it made a sound at all, would be a deep, soft whoosh, the sound of air moving through a cavern, or the sound of the sea pulling back from a pebbled shore. From the cytochrome B6F complex, the electrons are passed to another mobile carrier, plastocyanin. Plastocyanin is a small, blue copper protein that resides in the thylakoid lumen. It shuttles electrons, one at a time, to photosystem I. The copper ion at its core gives it a delicate blue color, a tiny jewel floating in the green, lit lumen. You can see it there, moving with a gentle, diffusive motion, carrying its electron like a small, precious gift. When it reaches photosystem I, it docks gently at the docking site, and the electron is transferred, quietly, smoothly, without any sense of strain. And now, the electron is at photosystem I. Here, it is energized by the absorption of another photon. Photosystem I has its own antenna complex, its own reaction center, its own special pair of chlorophyll molecules. The electron, carrying the energy it received from the first photon and now augmented by a second, is passed to another electron acceptor. Ferredoxin, a small iron, sulfur protein that resides on the stromal side of the membrane. Ferredoxin is a tiny, ancient protein, one of the oldest electron carriers in biology. It moves through the stroma with a quiet confidence, carrying its high energy electron to its final destination, the enzyme ferredoxin, NADP reductase. This enzyme, located on the stromal side of the thylakoid membrane, takes the electron from ferredoxin and uses it, along with a proton from the stroma, to reduce NADP+ to NADPH. NADPH is a large molecule, a carrier of reducing power, and it will be used later in the Calvin cycle to help build sugars. The formation of NADPH is the final step of the light reactions, the quiet, peaceful conclusion of the electron's long journey from water to this stable, energy-rich molecule. You can float here, near the ferredoxin, NADP reductase, and watch this reduction take place. It's a gentle chemical reaction, the transfer of 2 electrons and a proton to NADP+. The molecule changes shape slightly, becoming a bit more relaxed, a bit more energy, dense, and then it drifts away into the stroma, ready for its role in the Calvin cycle. Another NADP molecule takes its place, and the process repeats. It's a slow, steady production of reducing equivalents, the quiet accumulation of chemical potential. All through this chain, you can feel the rhythm. Electron in, electron out, proton pumped, proton accumulated, plastoquinone drifting, plastocyanin shuttling, ferredoxin carrying. It's a melody without sound, a dance without movement, just the quiet, reliable passage of energy from one form to another. There is no stress here, no competition, no deadlines. The electron transport chain operates at the pace of diffusion, at the pace of molecular collisions, at the pace of the light that drives it. And the light is always there, gentle and steady, never pushing, never demanding. You might notice, if you let your awareness expand, that the entire thylakoid membrane is a mosaic of these complexes. Thousands of them, millions of them, all working together in a quiet, coordinated rhythm. They are not synchronized in lockstep. Each one follows its own local pace, determined by the availability of light and substrates. But together, they create a steady, collective hum, the hum of energy conversion, the hum of life's most ancient industry. It's a sound you can almost hear, a deep, resonant OM that underlies the whole chloroplast, the whole cell, the whole leaf. And you are held within that hum. It vibrates through you, not as a disturbance, but as a soothing, grounding frequency. It's the sound of continuity, of a process that began long before you and will continue long after. A process that asks nothing of you except that you be present, that you rest, that you let the gentle energy of the electron flow carry you deeper into peace. Now, you can drift slowly away from the electron transport chain, following the path of the protons into the lumen, or following the NADPH into the stroma. Either path leads to quietness. Either path leads to the next phase of the journey, the peaceful rotation of ATP synthase, and the calm, cyclic dance of the Calvin cycle. But there is no rush. The electrons are still flowing. The protons are still pumping. The light is still glowing. And the journey continues. Always. Now, let your awareness drift toward one of the most beautiful and peaceful structures in all of biology, ATP synthase. This is a large enzyme complex that spans the thylakoid membrane, and it uses the proton gradient, the quiet accumulation of protons in the lumen that you witnessed earlier, to synthesize ATP. ATP is the energy currency of the cell, the molecule that powers nearly every active process of life. But here, in the chloroplast, its synthesis is a calm, almost meditative process. You can see ATP synthase from the stromal side. It looks like a mushroom or a tiny, delicate fountain. It has two main parts, a membrane, embedded portion called CF lung, and a headpiece that protrudes into the stroma called CF1. The CF norpor portion forms a channel through which protons can flow, and the CF1 portion contains the catalytic sites where ATP is made. The whole complex is a marvel of quiet engineering, a turbine that runs not on steam or wind, but on the gentle, inevitable flow of protons down their concentration gradient. You position yourself near the base of ATP synthase, in the thylakoid membrane. You can feel the proton gradient, not as a physical force, but as a subtle, pervasive sense of readiness. The lumen is rich in protons, slightly more acidic than the stroma. The membrane separates these two compartments, and the only way for protons to pass back into the stroma, the only way that is permitted, is through the CF-ton channel. So they do. One by one, protons enter the channel at the lumen side. And as each proton passes through, it interacts with a ring of protein subunits called the C. Ring. This ring sits within the membrane and rotates, slowly, very slowly, in response to the proton flow. It's not a fast rotation. It's a stately, deliberate turning, like a water wheel in a gentle stream. Each proton that passes through causes the C ring to rotate by a small fraction of a full turn. The energy for this rotation comes from the dissipation of the proton gradient, the gradual equalization of charge and pH across the membrane. It's a process of quiet relaxation, of moving from a state of gentle potential to a state of peaceful equilibrium. The rotation of the C ring is coupled to a central stalk, a protein shaft that extends up into the CF1 headpiece. As the C ring turns, so does the stalk, and the stalk, in turn, interacts with the catalytic subunits of CF1. CF1 is a hexameric ring, 6 subunits arranged around the central stalk. They don't rotate, they remain stationary. But the rotating stalk causes them to change shape, sequentially, as it presses against them. These shape changes alter the affinity of the catalytic sites for ADP and inorganic phosphate, and for the ATP product. What happens, in essence, is this: one catalytic site binds ADP and phosphate loosely, then closes around them tightly, bringing them together so that they can react. The reaction, the formation of ATP, occurs spontaneously when the substrates are held in close proximity and the right orientation. Then the site opens, releasing the newly made ATP into the stroma. And the cycle repeats. As long as protons flow, you can watch this happening. It's a gentle, rhythmic cycle of binding, closing, reacting, and releasing. It's like a slow, quiet breath in, bind, hold, react out, release. The stalk turns. The sites change shape. ATP molecules emerge, one by one, into the stroma, where they will join the pool of available energy. There's no noise, no jerking, no sudden movements, just the smooth, continuous rotation of the stalk, the soft conformational breathing of the catalytic sites. The rotation itself is almost mesmerizing. If you let yourself become absorbed in it, you can feel your own sense of time slowing to match the pace of the enzyme. The stalk turns, perhaps a few hundred times per second in real time. But in your imagined, expanded time, each turn takes many seconds, even minutes. You can follow a single catalytic subunit through its entire cycle. Empty, loose, tight, release, empty, loose, tight, release. It's a mantra of shapes, a visual lullaby. And all the while, the protons flow. They flow from the lumen, through the key, ring, into the stroma. They are not in a rush. They are simply following their nature, moving from a region of higher concentration to a region of lower concentration. It's a passive, effortless process, diffusion, the most fundamental and patient force in the universe. The proton gradient, which was built up so quietly by the electron transport chain, is now being dissipated just as quietly, and the energy released is captured in the phosphoanhydride bonds of ATP. You might notice, if you look around the stroma, that ATP molecules are drifting away from the synthase, ready to be used in the Calvin cycle. They are small, compact molecules with 3 phosphate groups linked in a chain. The bond between the second and third phosphate is the one that stores the most energy, and it's this bond that will be broken to power the synthesis of sugars. But for now, the ATP molecules are just floating, waiting. They are like small, charged batteries, quietly awaiting their moment. There is no hurry. The Calvin cycle will use them when it needs them, at its own pace. And the ATP synthase continues to turn, the protons continue to flow, the gradient continues to be maintained by the ongoing activity of the electron transport chain, which is still pumping protons into the lumen, still receiving electrons from water, still being driven by light. It's a beautiful, balanced system, a steady state where production and use are perfectly matched, where nothing accumulates to excess and nothing runs short. It's a state of peaceful equilibrium, a calm, self-regulating harmony. You can let your thoughts rest on that harmony. The chloroplast is a place of deep balance, where every process is coordinated with every other process, not by a central controller, but by the inherent properties of the molecules themselves. The light reactions produce ATP and NADPH. The Calvin cycle consumes ATP and NADPH and produces ADP and NADP+, which feed back into the light reactions. The oxygen that is produced diffuses away. The carbon dioxide that is consumed diffuses in. It's a cycle, a circle, an endless, quiet round, and you are in the center of that round, floating in the stroma, watching the ATP synthase turn. The green light bathes everything. The proton gradient is like a gentle, electric whisper. The rotation is slow, rhythmic, peaceful. You can let yourself drift into the rhythm, letting your breathing match the cycle of the synthase in as the site closes, out as the site opens, in and out, in and out. Time stretches. The world outside the leaf fades to a distant, unimportant hum. There is only this, the enzyme, the protons, the ATP, the light, and now, very softly, you can let the synthase continue its turning without your focused attention. You can carry with you the image of that slow rotation, the sense of effortless energy conversion as you drift toward the final part of your journey, the Calvin cycle, where the carbon dioxide that entered the leaf so long ago will finally be woven into sugar. But that can wait. The synthase still turns. The protons still flow. The light still glows. And the journey continues always. And now, you find yourself drifting deeper into the stroma, to the place where carbon dioxide meets its destiny. This is the Calvin cycle, a circular pathway of reactions that takes the CO2 that wandered in through the stomata, the ATP that was synthesized by the gentle rotation of ATP synthase, and the NADPH that was produced by the electron transport chain, and combines them to make glyceraldehyde 3-phosphate, a 3-carbon sugar phosphate that can be used to build glucose, starch, cellulose, and all the other carbohydrates of life. But you don't need to remember those names. You can just think of it as a quiet, cyclic dance, a round of reactions that turns and turns patiently, endlessly, weaving carbon atoms into the fabric of organic matter. It's a dance that has been going on for more than 2 billion years in the stroma of chloroplasts, in the cytoplasm of cyanobacteria, in the green tissues of every plant on Earth. And it's a dance that you are now invited to observe as a silent, drifting guest. The first step is carbon fixation. The enzyme Rubisco, that abundant, large protein you saw earlier, floating through the stroma like a soft cloud, takes a molecule of ribulose 140 bisphosphate, a 5-carbon sugar phosphate, and attaches it to a molecule of CO2. This reaction produces a 6-carbon intermediate that immediately splits into 2 molecules of 3-phosphoglycerate, a 3-carbon compound. The splitting is not a violent break, it's a quiet, inevitable hydrolysis, a gentle coming, a part that yields 2 identical fragments. You can watch this happen. Rubisco holds the 5-carbon substrate in its active site, a pocket of precisely shaped amino acid residues. The CO2 diffuses in, settles into the pocket, and the reaction proceeds. There is no fanfare. The carbon dioxide, which once floated lazily through the air spaces of the leaf, is now covalently bonded to an organic molecule. It has entered the world of life. It's a quiet moment, a small miracle that happens millions of times per second across the planet. And yet it is utterly calm, utterly without drama. The 3-phosphoglycerate molecules are then phosphorylated by ATP. They receive a phosphate group, becoming with 3-bisphosphoglycerate. This is a preparatory step, a gentle activation that makes the next reaction possible. The ATP, which you saw being made by ATP synthase, now gives up some of its stored energy, breaking its terminal phosphate bond and becoming ADP. The ADP drifts back toward the thylakoid membrane, where it will be recharged by the light reactions. It's a cycle within a cycle, a quiet exchange of energy and matter. Then, NADPH, the reducing equivalent you saw being produced by ferredoxin. NADP reductase donates its high energy electrons to 1003-bisphosphoglycerate, reducing it to glyceraldehyde 3-phosphate, or G-serine. This is the first sugar molecule of the cycle. It's a 3-carbon sugar phosphate, a small but crucial building block. the NADPH becomes NADP+, and it too drifts back to the light reactions for recharging. The G3P is now available for the next stage. Some of the G3P molecules leave the Calvin cycle to be used in the synthesis of glucose and other carbohydrates. Two G3P molecules can combine to form glucose-6 phosphate, which can then be converted to glucose, or starch, or cellulose. Starch is stored in the chloroplast as a reserve of energy. Tiny grains that you can see if you look carefully. They glisten in the green light like granules of pale sugar. Cellulose is exported from the cell and used to build the cell wall, the very fabric of the leaf's structure. In this way, the carbon that entered as a gas becomes solid, becomes the substance of the plant, becomes the food that sustains nearly all life on Earth. But most of the G3P does not leave the cycle. Instead, it is used to regenerate ribulose E-fulv bisphosphate, the 5-carbon sugar that Rubisco needs to keep the cycle going. This regeneration phase is a complex series of reactions that rearranges carbon skeletons using more ATP. 3 molecules of G3P, containing a total of 9 carbons, are rearranged to form 3 molecules of a 5-carbon sugar, containing a total of 15 carbons. It's a puzzle that the enzymes solve effortlessly, passing intermediates from one to the next in a quiet, coordinated relay. There is no hurry. Each enzyme takes its substrate, catalyzes its reaction, and releases the product, which diffuses to the next enzyme. It's a slow, orderly progression, like a line of people passing buckets of water from a well to a garden, each one moving with a calm, steady rhythm. You can follow the pathway if you wish, tracing the carbon atoms as they move through sugar phosphates with names like setoheptulose bisphosphate and xylulose phosphate. Or you can simply let the cycle turn in your awareness, a gentle wheel of reactions that never stops. The enzymes involved, transketolase, aldolase, phosphatase, and others, are all floating in the stroma, each one doing its small, specific job. They are like quiet artisans, each with a single, simple task, working together to create a harmonious whole. And as the cycle turns, you can feel the deep, cyclic rhythm of it. It's a rhythm that matches the rotation of ATP synthase, the flow of protons, the capture of light. All the processes you've witnessed on this journey are connected, like the parts of a single, great, breathing organism. The light reactions and the Calvin cycle are 2 halves of a whole, linked by ATP and NADPH, by the protons and electrons, by the water and the carbon dioxide. It's a unity, a oneness, that has existed since the first photosynthetic cell drew its first breath of light. You can let yourself rest in that unity. The Calvin cycle turns. The stroma is filled with the quiet hum of enzymatic activity. Rubisco continues to fix carbon. G3P continues to be made. Some of it becomes starch. Some becomes the cycle itself. The carbon atoms, once free and wandering, are now part of a living system, part of the great green tapestry of photosynthesis. And you are here, a witness, a quiet presence held within that tapestry. Time moves differently here. The Calvin cycle can take minutes, hours, days, depending on the needs of the plant. But in your experience, it's a timeless round. There is no beginning and no end. You can enter the cycle at any point and leave at any point, and the cycle will continue just the same. It's a circle, a ring, a quiet, eternal return. And that's a comforting thought, that some things have no conclusion, no final moment, but simply continue round and round in perfect peace. Now, as you feel yourself beginning to draw back from the Calvin cycle, you can take with you the image of that gentle wheel, the quiet weaving of carbon into sugar, the patient regeneration of the substrate, the steady, rhythmic flow of energy from light to matter. It's an image you can return to anytime you need to feel grounded, anytime you need to be reminded of the deep, slow, peaceful processes that underlie the busy surface of the world. And the Calvin cycle continues. Rubisco fixes another CO2. ATP synthase turns. The electron transport chain hums. Water is split. Oxygen rises. Light is captured. The leaf breathes. And the journey continues, always. And now, very gently, you begin to draw back from the chloroplasts, from the stroma, from the thylakoid membranes, from the Calvin cycle and the light reactions and the quiet, green glow that has surrounded you for what feels like a long, peaceful age. There is no abrupt ending here, just a soft, gradual receding, like the tide pulling back from the shore, leaving the sands smooth and undisturbed. You find yourself floating back through the cytoplasm of the mesophyll cell, past the cytoskeleton, past the other chloroplasts that still turn, still capture light, still fix carbon in their own quiet rhythms. The green light fades slightly, becoming paler, more diffuse. As you move outward, the cell membrane approaches, and you pass through it without resistance. Back into the air spaces of the spongy mesophyll. The air here is still cool and moist. The carbon dioxide still drifts. The oxygen still rises. The stomata, those gentle oval doorways, are still open, still breathing the slow, rhythmic breath of the leaf. You drift toward one of them, moving with the same patient, meandering motion that you had when you first entered. The guard cells are relaxed. The pore is wide, and outside, beyond the leaf, is the quiet, waiting world. You emerge, and you are once again resting on the surface of the leaf. The waxy cuticle is smooth beneath you. Above, the canopy of branches shifts slightly, dappling the light. The warmth of the sun is gentle, a comfortable blanket. You can hear, very faintly, the sounds of the outside world, a distant bird, a breeze stirring the leaves, but they are soft, far away, and they don't disturb you. You've traveled through the quiet expanse of a single leaf. You've drifted through air spaces and cytoplasm, through membranes and lumens. You've watched photons captured by antenna complexes, electrons passed along transport chains, protons pumped and flowing, ATP synthase turning, the Calvin cycle weaving carbon into sugar. You've witnessed the splitting of water and the rise of oxygen. You've been a silent guest in one of the oldest, most patient processes on Earth, and you carry the peace of that journey with you now. It's a peace that doesn't demand to be remembered in detail. It's a peace that settles into your bones, into your breath, into the slow, steady rhythm of your heart. The chloroplast still turns inside its leaf. The light still falls. The world is still green, still breathing, still quietly at work. And you are part of that green, breathing world held within it as you drift now towards sleep. The journey doesn't truly end. It circles back on itself, like the Calvin cycle, like the flow of protons, like the turning of the synthase. You can re-enter it at any point, any night, any moment when you need to rest. The leaf will always be there, waiting, The light will always be there, shining. The process will always continue, in its own time, in its own peace. And so, as you settle into the softness of your bed, as your breathing slows and your thoughts grow quiet, you can let the image of the leaf stay with you. The green light, the quiet hum, the slow turning, the patient breathing, nothing to figure out, nothing to solve, nothing to hold on to, just rest. The leaf breathes in, the leaf breathes out, and the journey continues, always. As you drift gently into sleep,